Hello and welcome everyone to this month's ZOMI webinar. My name is W60 Silva and I'm co-moderating our session today together with my colleague Pina Yilmaz. We would like to welcome our speaker, Professor Marco Düring, who is presenting today on the topic validation of automated image analysis tools in the absence of a ground truth. Marco is a neurologist by training with a scientific focus on developing neuroimaging markers for neurodegenerative and cerebrovascular diseases. He worked previously at the Center for Molecular Biology in Heidelberg, at the Institute of Stroke and Dementia Research in Munich, and at Radboud and Donders Institute in Nijmegen. In 2020, he joined the Medical Image Analysis Center in Basel as Chief Scientific Officer. And as a short reminder to our audience, before we start, please don't be shy and ask your questions by typing them into our Q&A box. We will address them at the end of the presentation. And without further ado, please, Marco, the stage is yours.
Yeah, thanks a lot for the introduction and for the invitation to this webinar. So before beginning, quickly the disclosures. These are not relevant for the presentation, but I'd like to mention that I'm an employee of MIAG AG, which is a neuroimaging CRO. So what I want to cover today is a short introduction to the general topic of imaging marker validation, then followed by two use cases. And the first being deep learning-based brainstem segmentation, and then the application of diffusion imaging. So first talk about the central topic of ground truth, defined as the true value of a variable or a phenomenon that is being measured or observed. Or to make it more concrete in the terms of imaging, for example, lesion segmentation, a lesion mask or label mask that is generated, for example, by an experienced and trained rater, so manual segmentation. And that is very often used as a synonym to a gold standard, which is more a point of reference or something that is determined by expert judgment, consensus. So by speaking about ground truth, I mean both rather broadly, and especially looking at cases where we lack these ground truth or gold standard. And even if we have a ground truth, there are still limitations. So very often they are, or this type of data is difficult to obtain. So if you think about lesion segmentation, this is of course very labor intensive and you need really trained experts to do this. It takes a lot of time and is thus also expensive. Using ground truth is fully suited only for a fraction or some tasks. For example, classification tasks. That's really straightforward, especially if you have neuropathological validation for disease and control, then this can be straightforward. But again, with lesions, also raters disagree. So what is actually the ground truth when segmenting a lesion? And by only using ground truth for validation, you actually, if an algorithm already exceeds the performance of a human rater, it can still, by definition, never be better. Because if you validate against the ground truth is by definition what is right. So validation in general, what is validation actually? What do we mean by that? Well, simply spoken, validation is a testing of requirements that you set. And in terms of technical validation, this is pretty straightforward, as you can see here. So for example, of course, you have the requirement that an algorithm or a variable or a measurement is repeatable. Meaning that if you measure it twice, for example, if you put a patient twice in the scanner, within short time, you expect to get the same result for your measurement from your algorithm. And of course, you also want your algorithm to be reproducible. So if you measure it on two different scanners, even a 3 Tesla and a 1.5 Tesla or a Siemens or a Philips, you also expect the same result. So that is your requirement. You want the algorithm to be reproducible. And you can design a test for that. The clinical validation, though, is not as straightforward. So how do you validate in a clinical sense? And that very much depends on the intended use. And I think this might be my most important slide. The first step in any validation is to clarify and think about the intended use of your algorithm or biomarker. So will it be a diagnostic biomarker? Do you want to separate patients from controls, or is it more for monitoring, for prediction, if a certain marker can predict whether a treatment will be effective? Or is it a prognostic biomarker? For example, predicting the conversion to dementia. Or is it safety? Well, all of this is defined in this BEST resource by the NIH Biomarker Working Group and the FDA. The link is down below here. But your task is really, if you start validation, to make up your mind what kind of marker are you actually validating. And for today, the use case or intended use that we are going to cover is the surrogate endpoint. So an imaging marker that can be used to determine the efficacy of a treatment and can possibly also replace a clinical endpoint for drug approval. So let's go directly into a use case and how a biomarker validation was performed for our deep learning-based brainstem segmentation. So you can see on the left an example. This is a segmentation algorithm that segments mesencephalon, pons, and medulla oblongata. And this was developed together with our partners at the University Hospital in Basel, Regina Schläger from the Department of Neurology, as well as the University Basel Department of Biomedical Engineering, the group of Philippe Catin. So this is an in-house developed custom AI tool that is built on multidimensional gated recurrent units. And when evaluating this against the ground truth, as you can see here on the right, we achieve quite a good agreement with manually labeled ground truth segmentations for the brainstem. So in that sense, this tool looks really good, but now let's go deeper into the validation. So again, we are looking at clinical validation, technical validation, and we will test requirements. But what kind of requirements? So I already said the intended use will be a surrogate endpoint that can detect treatment effects, so efficacy. And of course, what we want to detect is atrophy and how a treatment would modify brainstem atrophy. Now, how can we test this? How can we get to meaningful requirements so we can test it? And we did here carefully select a patient sample that allows us to formulate meaningful requirements. This is the PROMESA phase 2 trial. The principal investigator was Johannes Levin from LMU Munich. Of course, if you want to study atrophy, we need longitudinal data. So this was baseline and 1-year follow-up data. And it's only 20 patients, but that's because it was quite a really special sample of patients. It's patients with multiple system atrophy, either the pontine or the cerebellar variant. And what this allows us, because this is such a rapidly progressing neurodegenerative disease, is allows us to formulate a requirement. And that would be that over 1 year, we expect to detect atrophy in all cases, so in all 20 subjects. And that is our requirement for clinical validation that we can now test just by analyzing the longitudinal data. And this is how it looks like. On the left, you see the volumes over time, so baseline and 1-year follow-up. And the first column is our algorithm, followed by benchmarking against 2 other algorithms. That's the state-of-the-art free surface subsegmentation pipeline, either longitudinal or cross-sectional. And we've done this for mesencephalon, pons, medulla oblongata, and brainstem. And here on the right, you can see the results expressed as % volume change over 1 year. And there's some interesting observations. So the first one is here, this clear outlier. So this is a patient where our algorithm actually detected a 15% volume increase in the medulla oblongata. Now, this was a clear segmentation error, and this is a typical phenomenon for deep learning-based algorithms. They usually work really well, and when they fail, they tend to fail in a spectacular way. So with a really clear outlier, but the good thing is that this is easy to spot for a human rater doing quality control. So I think it's quite crucial here to do, again, well, use AI tools, but do human quality control to spot these obvious errors. And now if we go to the primary readout, which is brainstem % volume change, you can see that indeed only our algorithm detects atrophy in 20 out of 20 cases, while in the benchmarking state-of-the-art algorithm, there are 2 to 3 to 4 cases that still show a volume increase. So in that sense, this ND-GRU algorithm met the requirement that we want to detect atrophy in all cases. So we can move on to technical validation. Here we used two data sets. One was acquired in Nijmegen at the Donders Institute, the so-called Run-DMC Intense study. This comprised elderly participants. They were enriched for small vessel disease, but they are very representative for an elderly study population. So for example, for a typical study in neurodegenerative patients. And that's 50 participants that were scanned monthly for up to 10 scans. So we call this high frequency serial imaging, and that allows us to do some nice things that I will show you in a minute. And the second data set is the MARC VCRD consortium. That's an NIH-funded consortium in the US, coordinated by MGH in Boston. Also elderly participants, also enriched for small vessel disease. And they have two sub-studies, a scan-rescan sub-study with 42 subjects scanned back-to-back within the same scanner, and an intersite sub-study, and that's 20 subjects that traveled to 4 different scanners. 2 Siemens, 1 Philips, and 1 GE. Now, let's first start with the Run-DMC Intense study. And our requirements here are very simple, that our measurements should be both repeatable and reproducible across scanners. And that is the high-frequency serial imaging data for repeatability. So 1 line is 1 patient and 1 dot is 1 scan. And again, we have in the column here the deep learning-based method, and then for benchmarking, FreeSurfer in the longitudinal and the cross-sectional method, and now showing only mesencephalon, pons, and medulla oblongata. You can again clearly spot some outliers, this time for the FreeSurfer pipeline, but also here in the deep learning-based pipeline. But how to quantify now this? How do we get meaningful results and can compare it with so many measurements, monthly measurements? Well, one method is to look at the so-called limits of agreement of the mean. And this is shown here. It looks a bit like a Bland-Altman plot that you might be familiar with. And in essence, we're looking at the 2 blue lines, and the more narrow they are, the closer to 0 they are, the more repeatable the measurement is. And what you can see is that the deep learning based method now here shows overall a very good, compared to the benchmarking methods, very good repeatability that is either better or comparable to the other methods. And what this high frequency data allows us is not only to look at the repeatability, but also to learn more about outliers. If you make the assumption that atrophy is a linear process over time, over these 10 measurements, then we should indeed see, this is exaggerated here, but a slow decline over time. And ideally, if it's very good in terms of repeatability, we have a low variability, so all points should line up along this line. So in participant 1, we would have here 1 outlier, and participant 3, for example, has overall a higher variability, has multiple outliers. And we can look at this in 2 ways. One is at the participant level, the variability, so patient 1 or participant 1 would have a low variability, 2 would be medium, and 3 would really have a high variability. And then we can identify predictors for this variability. And you could imagine that, for example, age or disease severity might be a predictor for higher variability on the participant level. And then we can look for scan levels, so of these deviations from the, let's say, ideal point, or these basically residuals from linear fit. And again, find predictors, for example, motion in the scan or certain artifacts that do not occur on the participant level, but only on the scan level. So this allows us to dive deeper into technical validation and find predictors for variability. Okay, let's move on to the MARC VCRD dataset. So this was collected in a consortium, NIH funded in the US, and as I said, coordinated by the MGH in Boston. And this is repeatability, now a classical scan-rescan study, but in 42 subjects, and presented here as classical Bland-Altman plots. So also here you expect to be around 0, so to see no bias here with this line, and to have narrow limits. And this is the case here for this deep learning-based algorithm compared again to the benchmark. So it's either better or comparable to the benchmark method. And FreeSurfer, you might know, calculates for several hours. With this deep learning-based algorithm, we get a result within a couple of minutes. Also looking at the reproducibility. So here it's shown a little bit different. It's trio scanner versus Prisma scanner, so 2 Siemens 3 Tesla, and then those 2 scanners against the Philips Achiever. There's also GE scanner in this data set, but unfortunately the field of view was too narrow, so the medulla oblongata was cut off the scan, so we were not able to analyze the brainstem segmentation with the GE data. And what you can see is a fairly good agreement between the different scanners. The methods, I would say, are all roughly equal, maybe a little bit better here for the deep learning-based method. However, there's also a little bit of a bias as visible, so it's shifted from 0. But overall, I would say good reproducibility. And for that, I would like to move on to the second use case, which is diffusion MRI. And here, of course, it gets even more complicated because what is a ground truth for diffusion MRI? It's a quantitative MRI method that quantifies water diffusion in the brain, but we don't have any ground truth, and it's extremely difficult to obtain even post-mortem with pathology. If you look at a post-mortem brain, the water exchange that happens during death, the degradation after death, if you then fix the brain, so before the pathology looks at the tissue, it's no longer a ground truth for diffusion MRI, really. But can we still validate it for use? Of course. So the common diffusion models, I'm pretty sure you know diffusion tensor imaging. Here's some fractional anisotropy imaging, but this is, of course, only one of the diffusion models. There's also free water imaging, for example. It's a B-tensor model, or higher order diffusion models like cortosis that require multi-shell diffusion data. There's also, you might have heard of NODI, a biophysical model that really tries to model the architecture of the brain and biophysical properties. So many diffusion models, and really a lot of work has been done to validate diffusion MRI, for example, for memory clinic patients. So patients that visit the clinic because of memory problems or other cognitive problems. And we and others have done some work both for clinical and technical validation, and I want to give you a short overview of this work. What is typically seen is quite a strong correlation between diffusion-based markers and cognitive deficits. For example, here, 64% of explained variance, that's really high. But does it validate something? If you think about our intended use, does it validate one of these uses? Not directly, at least. But there are other studies, for example, for using diffusion-based metrics as a prognostic marker, predicting the conversion from MCI to dementia. And here you can see, and that was a requirement, that diffusion imaging really adds predictive value or prognostic value better on top of only clinical variables, for example. So in this study, this requirement was fulfilled for clinical validation. Now, if you think again of a surrogate endpoint, so an efficacy measurement for clinical trials, our requirement would be that the sample size we need when using an imaging marker is smaller than the sample size that we need to see an effect with clinical measures. And this is shown here. So this was a study published in 2016 already. We simulated a hypothetical clinical trial from longitudinal data, 30% treatment effect, meaning a 30% slowdown of disease progression or imaging marker progression within 1.5 years. And you can see if you use neuropsychological assessment in these patients, you would need to detect the 30% treatment effect more than 5,000 participants. And this goes down, and with an imaging marker based on diffusion imaging, that's only 100 patients. So that requirement for clinical validation was also fulfilled. We can reduce the sample size when using a diffusion imaging marker. Technical validation in these markers, this is a study from Pauline Maillard at UC Davis. And here you can see for the marker free water now a really good scan-rescan repeatability, as well as excellent interscanner reproducibility, maybe a little bit of a shift between the Siemens and the GE scanner, but overall very good reproducibility. And this is also what we have seen in our data. Again, you can see here the high frequency imaging, the RunDMC Intent study with these 10 scans. And again, one patient is one line. 5 of them are just highlighted so you can better follow them. A really stable marker over time. This is now cortosis. And we also did some reproducibility with some interesting findings. So here, for the tensor model between a Prisma scanner and a Skyra scanner, completely different gradient systems, which are actually crucial for diffusion imaging, you find almost a perfect agreement between these two scanners. But for the NODI model, which is a higher order model, a more complex model, modeling intracellular compartment, extracellular, and free water, you can see a certain shift from the ideal. So the ideal would be the black line, and it's a little bit shifted. There's a certain bias. And that's an interesting observation, that the simple model has a almost perfect reproducibility, whereas the more complex model shows some deviations. And that is a common theme in diffusion imaging. I mean, you make so many arbitrary choices in diffusion imaging, starting with your raw data, how many diffusion directions, resolution, what model to fit. Is it a tensor model or a more complex model like constrained spherical deconvolution? Do you do tractography? Then you have even more arbitrary choices, tractography algorithms, stop criteria, where to seed the tractography. Is it even a conic tome analysis? And yeah, these are all the things that you should actually validate if these choices have an impact on your metric. And what you also should at least consider is, what are you actually measuring now, especially with such a rather unspecific method like diffusion MRI. And given that for the use case dementia patients, that almost all dementia patients, this is neuropathological data from Chicago, from Julie Schneider, even if they have been clinically diagnosed with Alzheimer's disease, you rarely find only Alzheimer's disease. That's only 3% of patients in neuropathology. You mostly find a combination of Alzheimer's disease pathology, vascular pathology, and even a third degenerative pathology, or third pathology, a second degenerative. So that is the vast majority of cases. They have mixed pathology. And what are you actually measuring with diffusion MRI? Well, we have good reason to believe that is actually the vascular contribution. And I don't have the time to dive more into that, but just as a train of thought, it's mostly, diffusion MRI mostly picks up the vascular pathology in these patients. So let me summarize. For biomarker validation, for imaging marker validation, in my view, the most important thing is to clarify the intended use, because then you can come up with meaningful requirements instead of just using a ground truth and looking, for example, to the overlap with ground truth segmentations. You need carefully selected samples for both clinical and technical validation, because you want, of course, to validate your method in the target population, not in young, healthy subjects, not in your co-workers, not in young, healthy PhD students, but in patients. For example, memory clinic patients, if this is a marker that you want to apply in a memory clinic population. And also populations that allow you to define meaningful requirements. For example, these MSA patients that we use because of their rapid progression, that allow you to have this requirement that you want to detect atrophy in all of the patients. And keep it simple, especially I think if you use more quantitative, more advanced imaging methodology, at least that is our experience. For example, it's very simple tensor model outperforming clearly in terms of technical validation, the more complex biophysical models. And with that, I would like to thank all the collaborators in this study. That's our colleagues in Basel, in Munich, in Nijmegen, and in the US, the Mark VCRD Consortium. Yeah, and thank you very much for your attention, and happy to discuss with you now.
Thank you very much, Marco, for this great overview. And yeah, it's very exciting what you've shown so far. Especially we also worked a bit on the PSMD marker. I have a few questions. I don't see any questions yet in the Q&A box. So first I want to, the intriguing graph what you showed on the more complex model, because in the end, depending on, of course, what you greatly explained about what kind of marker are we looking at? So what is your intention? And then what does this marker mean in different populations? I mean, also from an epidemiological perspective, you want to know what it in general means in a general population. Also, what was interesting for the PSMD marker, and then to target it well, to see how we can use it in specific targeted populations. So my question is, in the end, for a marker to have a meaningful purpose, you want to model it in a complex way, right? So how do we solve the deviations as what you showed us in the NODI? So I was curious on how did you explain the deviations within that graph? And what are tools that we can think of in when we pursue research?
Absolutely. I think this is a hot topic in the community. I can only speculate about the reasons why we saw this shift. It probably has to do with the different gradient system, although we selected, let's say, the same gradient setting on both scanners. So they should be roughly equal, but it should be the gradient system, and probably resulting differences, minimal difference in also echo time. I mean, you can carefully harmonize and standardize your protocol, of course. You can think of even after acquisition of harmonizing data using modern algorithms. Most of them, though, at least the more sophisticated algorithms, require that you not only have your study sample, but you also acquired healthy controls on all scanners. So there are methods to correct this. In the end, I think if you apply something that is more complex, you should also be sure that it has an added value. It's always a trade-off. More complexity usually means less technical validity. So you should really make sure that you have also a benefit out of it. And if you have no benefit, there's no reason to use the more complex method.
Thank you. So in the end, it actually distilled to the technical part, which is of course great in the harness consortium as well, to harmonize the data in partially, if it's possible, in acquiring, but also in the post-processing, I guess.
Yes. And I think there's still a great difference, of course, between a single center study and a multi-center study. And then a multi-center study, let's say in centers with very modern MRI machines, and in the real world, maybe also including developing countries. I mean, there are many levels of, let's say, harmonization that you can achieve or cannot achieve, and you have to just adapt your methods accordingly.
Great, thank you. Then another thing about what are we measuring? So, I mean, for example, going back to the use case that you showed, especially the diffusion, what are your main also take-home messages in the measurements? In where should we start with the use case?
Yeah, I mean, in terms of what are we measuring, I think the most crucial part is to always remind ourselves that MRI is inherently unspecific. That is true for diffusion imaging. That is also true for, let's say, myelin imaging, even magnetization transfer, myelin water, you name it. These are not fully specific for a certain thing that we measure, and we have a summary measure out of a huge volume, even if it's 1 millimeter isotropic. I mean, there's so many things in there. I think that's the most important part. And then, of course, you can try to tease out certain mechanisms, maybe also in combination with a fluid marker or with PET imaging, for example. So I think it's the beginning. We need multimodal analysis, combining not only clinical values and imaging, but also biomarker studies, PET imaging, to further understand what we are actually measuring. Is it important for every use case? So is it important, for example, for a diagnostic marker to know what are we measuring exactly when it can perfectly differentiate between a patient and a control? Probably not. Mm-hmm. If it can predict dementia on top of clinical measures, maybe I don't care what I'm actually measuring. I just want a marker that can do this. But still, to keep it in your mind, right? We don't actually know what we're measuring, but it works. It's a fine approach, I would say.
Thank you. Yeah, because it's regarding what you showed with the free water. That's now the next hot topic, I guess.
Mm-hmm. Free water, I mean, is also pretty unspecific in the end. It's certainly driven by small vessel disease. I think we've shown this quite clearly. It might be driven by inflammation, but it might also be driven by demyelination and axonal degeneration. In the end, it's a mix, and it might very much depend on where you measure it in the brain and in which population, if you measure more a vascular effect or more an inflammatory effect. I think it can be made more specific if you apply it in a certain way, but it will never be fully specific.
I think it's also again, thinking of what kind of purpose you have. I think the way towards it is to combine, especially what you said, the clinical and also the lab, if we are already there, and then the imaging to have the complete overview. These were the questions I had for you. Do you still have questions, Vera?
Yeah, I might add one question. I haven't seen our Q&A box. It's still not very full.
It was very clear.
Yeah, I think so. I would like to have your comment, Marco, on the necessity of a human reader to do some kind of quality assessment, quality control of the output. What do you think? This is just for this outliers that we suspect is an error, or do you think we have a bigger role in the quality control?
I think that is really from case to case can be different. But this should be an integral part of any validation study. I mean, to already think about how does the human reader, the radiologist, come into play. I mean, the minimum, I think, for every method is a QC to see if it makes sense to sort out gross errors. That could maybe even done by trained raters, doesn't need a radiologist. But for many use cases, there's for sure still a radiologist needed to check the output, if it makes sense, if it's plausible. I think in the near future, this is very much mandatory, and it should be really a part of every validation study to already think about how to integrate the human reader into the whole workflow, and to also include that in the validation, actually.
Yeah, I think regarding the visual inspection, right, for I think almost every scan, you have to have the major things rated, at least by some kind of a rater, to be able to put the images in the algorithm as well. Because I mean, for great, I know for the certain tools I've worked with for great infarcts, etc., it's not always working correctly, and for calculating volumes, for example.
Yeah, I mean, every model, also deep learning-based models, have assumptions. So if the assumption is I see a brain of a dementia patient without a stroke, then it will only work in this population if it's trained in that population. If you violate an assumption, of course, the model falls apart. So that is actually quite important to know the assumptions or the training data and to check for those. Absolutely. Yeah.
Thank you very much. I think we're done with our questions. Thank you again for joining us for this great evening with great presentation and answering all of our questions. And I also want to thank the audience for joining us. And I would like to remind everyone that we have our annual meeting this year in Pisa on October 13th and 14th, and we will see you at our next webinar. Have a great week, everyone.
Thanks a lot. Bye-bye.
Thank you. Bye.
Okay.